without question, the most recent development that was, I think, a large surprise to the field was the important role of Bcells in disease, in induction. We make different animal models to try to teach us about the disease, but those models are not terribly good for teaching us what causes the disease, but are better for telling us what are the consequences of activated T cells. So the clinical trial, which led to B-cell depletion with rather remarkable results, to me is perhaps the most important recent discovery in MS. It's all about the clinical trial at the end of the day. Many of us who are immunologists have come to the conclusion that you need to treat MS very aggressively, very early. And if one does that, it will perhaps prevent secondary progressive disease and neurodegeneration. So the question of whether early treatment will indeed prevent progressive disease is extremely important. Another really unmet need is the question of how long does one need to be on treatment? For example, with anti-B cell treatment, does one need it for 2 years, 5 years, 10 years? Is there a way of stopping the treatment after one begins it? Can one re-zero the immune system? I think the other really big discovery in MS is the identification of the genes that cause MS. We had our International MS Genetics Consortium meeting here on Tuesday before the meeting began, and our work, which hopefully will be published shortly in Science, we've identified 233 genetic variants associated with the risk of developing MS. So using those data, it's taught us that MS is both a T cell and B cell disease, and is pointing to pathways that we may be able to target. Well, from my own perspective as an immunologist, the most promising areas relate to new technology called single-cell sequencing, RNA-seq. We now have the ability to take cells directly from the spinal fluid. We can even take antigen-specific cells directly from the spinal fluid and to actually sequence them at a single-cell level. We've never been able to do that before. Presented some of those data yesterday. So it means we can take T cells that we think are actually causing MS because they're reactive to myelin. There may only be a dozen of them in the spinal fluid, but we can actually see what they are functionally and what pathways we're using. So to me, it's exciting new technology which is invading the field of medical science, which should give incredible insight into the cause of the disease. Well, to be perfectly honest, the debate I had with Professor Steve Hafler, our Rumble in the Jungle, where the two of us, I think, were set up to have a debate about which is more important, T cells and B cells. We didn't know what each other would really say, but it was bemusing that we both ended up saying the same thing. So that, on a personal level, that was the most interesting to me. There have been a number of other interesting talks. It's a wonderful time to be taking care of MS patients. When I began in the 1970s, we had no treatment, just steroids. So a patient would come in and we'd say, go home, there's nothing we can do. We've gone from that to incredible therapies that really appeared to change the course of the disease. So from a clinical perspective, it's really an amazing time to be a physician and being able to care for MS patients. From a scientific perspective, we now have the genetic architecture of the disease elucidated. That's really new. We have, as I mentioned, these new technologies for understanding the disease. And I look forward to the next generation of physician scientists continue to attack the disease and hopefully understand the nature, not just of relapse-remitting disease, because at one level I think we have a good working model for what causes MS. And it's probably true based on the clinical trials. We don't have as a good working model for progressive disease. So the gauntlet's been thrown down to the next generation to figure it out.